The research article we selected for our presentation is titled Chlamydia trachomatis polymorphic membrane protein D is a virulence factor involved in early host cell interactions, authored by Dr. Laszlo Carey and other researchers in 2014. This research study was conducted at the Laboratory of Intracellular Parasites at the National Institute of Allergy and Infectious Diseases, or NIAID, which is a branch of the National Institutes of Health, or NIH. This study was done in Dr. Harlan D. Caldwell's lab. As of April 2020, Dr. Caldwell is the chief of the Chlamydial Diseases section at the Laboratory of Clinical Immunology and Microbiology at NIAID/NIH. Dr. Carey, the first author of this study, is a research fellow at NIAID/NIH. This paper was published in Infection and Immunity, a medical journal published by the American Society for Microbiology. As of 2017, the impact factor of infection and immunity was 3.256. Chlamydia trachomatis is an obligate intracellular mucosa-tropic pathogen that causes blinding trachoma and sexually transmitted infections. C. trachomatis has a polymorphic membrane protein D called PMPD, which is a highly conserved autotransporter in the C. trachomatis genome and is a target of broadly cross-reactive neutralizing antibodies. The authors created a PMPD null mutant and showed that the PMPD gene is not an essential chlamydial gene, but is a virulence factor. They showed that the mutant attenuated infection for cultured human cells and reduced infection for human endocervical cells. Pictured here is C. trachomatis in a HeLa cell. The chlamydia is in green, whereas the F-actin in the HeLa cell is red and the nucleus is in blue. Determining pathogenesis has traditionally been limited by the lack of genetic tools. However, newly developed genetic approaches are enabling researchers to determine specific chlamydial genes C. trachomatis uses for pathogenesis. A gene of interest is PMPD, which is one of 9 autotransporters encoded by C. trachomatis. Autotransporters are part of gram-negative bacterial type 5 secretion systems, which are known for having a simple self-sufficient export mechanism, hence the name autotransporter or self-transporter. Autotransporters are also known to be virulence factors that aid in host cell interactions and host immunity evasion. PD is known to be conserved across C. trachomatis strains located on chlamydial surfaces and a participant in immune evasion, but its role in pathogenesis is not completely understood. The authors set out to determine if PMPD is an essential chlamydial gene and if it is a virulence factor. To investigate this, the authors created a C. trachomatis PMPD null mutant. Chlamydial infection begins with the attachment and invasion of vaginal or conjunctival epithelial cells by infectious, non-replicating chlamydial elementary bodies, or EBs. Once inside the epithelial cells, the EBs differentiate into non-infectious replicating reticular bodies, or RBs. The RBs will then migrate towards the nucleus and form an inclusion near the nucleus in order to replicate. Here, the epithelial cells can become latent if they are under stressful conditions, or the RBs differentiate back to EBs. Once EB differentiation is complete, the epithelial cells will either release the EBs via extrusions or are lysed. The entire process takes 2 to 3 days. Chlamydia traditionally presents as a sexually transmitted infection. Most people are asymptomatic, with only 10% of men and 5 to 30% of women showing symptoms in clinically confirmed chlamydia infections. Both men and women present with abnormal discharge and a burning sensation during urination. Women can develop cervicitis, urethritis, and pelvic inflammatory disease. Men report urethritis, and epididymitis, resulting in pain and swelling in one or both testicles. Chlamydia can also infect the rectum, causing rectal pain, discharge, and bleeding. Certain subtypes of C. trachomatis can also cause trachoma, a contagious chlamydial infection that affects the eyes. Here, the infection can be spread through contact with eyes, eyelids, or nose or throat secretions of infected people. Symptoms of trachoma include mild itching and irritation of the eyes and eyelids, discharge from eyes containing mucus or pus, swelling of the eyelids, light sensitivity, and eye pain. If not treated immediately, repercussions include eyelid scarring, ingrown eyelashes that can scratch the cornea, clouding in the cornea due to inflammation, and partial or complete vision loss. In this study, authors wanted to figure out whether or not PMPD played a role in C. trachomatis' ability to cause ocular infections. Authors began making the PMPD null mutant by placing McCoy cells in a 96-well tissue culture plate and infecting them with 10 IFU of chlamydia. IFU stands for inclusion forming unit, and it is a way to determine the amount of chlamydia present in a sample. The McCoy cells were infected for 48 hours. The chlamydia was collected and used to infect more McCoy cells another 2 times. McCoy cells are murine fibroblasts, which have been used as the standard cell line to study chlamydial infections. The chlamydia was retrieved from the McCoy cells, its DNA was extracted, and PCR was used to amplify PMPD. PMPD was then processed and visualized with gel electrophoresis. The mutants were sequenced, and those containing nonsense mutations like the one shown in the figure on the right in red were plaque cloned 3 times. The mutant genes were then propagated in McCoy cells and purified. Next, the conjunctival surfaces of both eyes in male and female cynomolgus macaques were directly inoculated with C. trachomatis. 20 microliters was administered to each eye to achieve 2 × 10⁴ IFU of chlamydia per eye. The PMPD null mutant C. trachomatis was administered to 6 of the macaques, and another 6 macaques received the wild-type strain. Vets performed clinical evaluations of the ocular disease weekly. The extent of the disease was determined based upon presence and severity of hyperemia and the number of follicles formed. Authors used macaques because C. trachomatis is species-specific. No infection was observed when they initially used murine models. In this study, hyperemia refers to an increased amount of blood vessels in the eye, usually resulting in increased redness. Severity of hyperemia was calculated in the following manner: 0 for null hyperemia, 1 for mild hyperemia, and 2 for severe hyperemia. Severity of subepithelial conjunctival follicles were calculated as follows: 0 for no follicles, 1 for 1 to 3 follicles, 2 for 4 to 10 follicles, 3 for greater than 10 follicles, and 4 for follicles are too numerous to count. The pathology scores of the right and left eyes were added up, the maximum score being 12. Vets took swabs of the conjunctiva, and the recoverable IFUs were cultured on HeLa cell monolayers and enumerated. The macaques' chlamydial IFU and pathology scores were observed for 100 days after infection. The positive control in this experiment was the wild-type C. trachomatis strain. There was no negative control. From this experiment, authors gathered the following data. For Figure 4a, the y-axis shows the number of chlamydial cells present after weekly swabs from the conjunctiva were cultured in HeLa cells. This was done over the span of 100 days, shown here as weeks on the x-axis. For Figure 4b, the y-axis shows the clinical disease score. The score was determined using the hyperemia and subepithelial conjunctival follicle scores I discussed in the previous slide, and also measured over 100 days, shown as weeks on the x-axis. From this data, authors discovered that the PMPD mutant strain showed a significant decrease in infection compared to the wild type. The mutant strain was still able to survive and cause ocular disease in the macaques, suggesting that PMPD is not an essential gene for C. trachomatis survival. PMPD likely functions as a virulence factor. Both the IFU values and clinical disease scores for PMPD null mutant strain are several folds lower than the wild type, suggesting that the mutant strain is less virulent. The loss of PMPD in the mutant decreased C. trachomatis' ability to proliferate within the host, which likely indicates that the gene increased virulence. The authors then wanted to determine if PMPD was involved in infectivity and virulence of chlamydia in human endocervical and conjunctival cell lines. To determine this, human endocervical cells A2EN and human conjunctival cells HCJEN were used. Chlamydia, when sexually transmitted, infects these endocervical cells, whereas infections in the eyes occur in the conjunctival cells, which is why they use these two cell lines. They also use McCoy cells as the infectious control. Looking at the diagram, the blue, orange, and green circles represent endocervical, conjunctival, and McCoy cells, respectively. Each of these cell lines were treated with the PMPD null mutant or wild-type strains. All cells were then infected with 15,000 IFUs of chlamydia. Cells were either fixed after 36 hours post-infection or recovered in McCoy cells to count the IFUs of chlamydia. As mentioned previously, the McCoy cell line is used to study chlamydial infections as it is very easy to cultivate and quantify chlamydial units in these cells. To detect the infectious unit in McCoy cells, inclusion bodies are detected through immunofluorescence. In this assay, an antibody that is conjugated with a fluorescent dye can bind to inclusion bodies. The fluorescently labeled inclusion bodies can then be observed under the microscope for quantification. After fixing the cells, the authors graphed the percent of chlamydial IFUs in each cell line. On the x-axis are the different cell lines. The black bars represent chlamydial burden as IFUs from cells that were inoculated with the wild-type strains, and the white bars represent the chlamydial burden from cells that received the PMPD null mutant strain. On the y-axis, they graph chlamydial IFUs relative to the percent of the control, which means that they were comparing the white bar to its corresponding black bar. We see in our experimental conditions that when the endocervical and conjunctival cells were given the PMPD null mutant strain, the IFUs of chlamydia were reduced by 70%. In McCoy cells, however, after receiving both wild-type and PMPD null mutant strains, it resulted in the same amount of chlamydial IFUs. The graph suggests that the PMPD null mutant reduces the ability to infect endocervical and conjunctival cell lines. It verifies that PMPD is a virulence factor and may be used to attach to host cells and invade.